important lecture series of Indian Council of Agricultural Research. And today we have the lecture number 61 on a very important topic. The topic for the relevance in today's time, and that is the accelerated breeding by de novo domestication of the crop plants. The talk is being given by Dr. Gotz Hensel, who is the head of the Center for Plant Genome Engineering at the Institute of Plant Biochemistry of the Heinrich University in Düsseldorf. Mr. Hensel, I can just tell you it's a very important lecture series. And as I told you, this is the 61st lecture out of the 75 lectures. In the past, we had lectures of many eminent persons from many CGIAR institutes, from the World Bank, from many universities. So it's a very important platform because this 75 lecture series is being undertaken for a very noble cause, and that is to celebrate the India's 75 glorious years of independence. So thank you very much for joining for this important lecture series and for agreeing to deliver the talk on a very important topic. Friends, let me just introduce Mr. Hensel. He is the head of the Center for Plant Genome Engineering at the Institute of Plant Biochemistry of the Heinrich University in Düsseldorf. His main focus at the center is on targeted genome modification of plants. This includes classical transgenic approaches, but also the use of new methods of targeted mutagenesis, which is better known as CRISPR-Cas technology. The aim will be to establish and develop suitable vector and tissue culture systems for plant species that are in the research focus within sapless, which will enable genetic transformation and thus genome modification. Before he joined the center of this Düsseldorf, he worked for 20 years at the Leibniz Institute of Plant Genetics and Crop Plant Research at Gotz, Lebanon. There, he developed efficient transformation protocols for cereals, especially barley and wheat, and was involved in the discovery of a system for host-induced gene silencing of pathogenic fungi. After the discovery of the targeted mutagenesis methods, TALEN and CRISPR, he immediately tried to adopt these systems for crop species. Using these methods, he was able to contribute to a variety of plant research questions such as spike architecture and chloroplast development. He is particularly interested in the production of recombinant proteins in cereal grains, also known as molecular farming. Very interesting biodata, interesting contribution of Mr. Hensel. Mr. Hensel, I can just tell you this platform is being joined by very senior researchers, our vice chancellors of the agriculture universities, our senior faculty from the Indian Council of Agricultural Research. It is being live streamed. Our students are also connected to this. And then many more persons will get benefit from your talk. You will have around 40 to 45 minutes. And then if you permit, we can have few questions from the audience towards the end of this. So thank you very much. And the platform is yours now. You can take floor. Thank you. And thanks to Sunil for introducing this speaker. Yes, Hensel, please.
Many thanks for this very warm welcome and this kind introduction. And I'm well aware of the importance of this lecture series. And I'm very proud that I be part of this. And is it now? Yes. Okay. So I think you can see it now in presentation mode?
Yes, clear.
Okay. So as you already heard, so I'm working at the Heinrich University of Düsseldorf, and I'm the head of the Center for Plant Genome Engineering, as already indicated. So you might be also aware that in Germany we have a cluster of excellence in plant science, which is shortly abbreviated C+, and I'm also belonging to this. So this cluster includes the University of Düsseldorf, the University of Cologne, the Max Planck Institute in Cologne, and the research center in Jülich. And so this is the only excellence cluster in plant sciences which we have in Germany. So I'm also affiliated to the Palacký University in Olmütz, so in the Czech Republic. And yeah, the title is already given here. And so to start with, I will just briefly introduce with some more words, as you already heard, what I'm doing. So this is the place I'm working. This is the new building where the biology faculty recently moved in. And as you heard, I'm working on transgene technology development, molecular farming, and chloroplast development. So here down, you see the plants my group is working with. So as you already heard, I have a long-lasting history in cereal transformation. So that's why I'm still working on barley. I'm working on corn or maize. Recently, I started a project working on sunflower and another grain cereal, amaranth. So as I belong to the Institute of Plant Biochemistry headed by Professor Andreas Weber, and he is interested in photosynthesis, so that's why my interest is now also towards Moricandia and Talinum, as these species have special photosynthesis phenotypes. So one is a calm plant and the other one has C3, C4 intermediates. And of course, I'm using the protein model plants like tobacco and Arabidopsis. So some more words about development of transgene technology. So you might be aware that there are differences for monocots and dicots. And so here you can see different explants which we routinely use. And so we have established protocols for using these diverse explants for the different types of plant species we are working on. So then you might be aware that a constitutive overexpression is not always useful. And so that's why I'm also interested in developing expression systems which are either just color specific, because this might be beneficial for the transformation process, or as I'm working with cereals. So here, of course, endosperm specific expression systems are highly versatile. So as illustrated here by using the green fluorescent protein, so you can restrict the accumulation of your protein of interest just in the endosperm and not in the embryo. And here is also a deeper look inside of the cells. And of course, we are developing synthetic promoters combining different features for a specific expression of our transgenes. So then you briefly heard that I'm also interested in molecular farming. So molecular farming is the production of recombinant proteins in plants. And so there are diverse platforms, but as I'm working with cereals, so the cereal grain is a natural bioreactor, which can be easily store proteins at ambient conditions without degradation. And so that's why I thought developing such a system is very interesting. And here we collaborate with a company which is called Off Genetics, and it's based in Iceland. And they are doing on a commercial basis the production of recombinant proteins, in this case, epidermal growth factors, for example, which they then use in cosmetics. And so here, this relies on the expression of the recombinant protein in the barley grain, and then we extract those things. And in my group, Pune Poramini has a project on doing this because we want to compete with the yield. So as you want to produce a valuable protein, of course, the yield is always of most importance. And as this product is very well established, so together with Nico Dismayer from the University of Osnabrück, we develop a system which will accumulate much more, but I do not have time to go into much detail. Last but not least, so I'm also interested in chloroplast development. And so here I have a long-lasting collaboration with Karin Kropinska from Kiel University, who discovered or is interested in plant protein, which is Barley1, which is dually localized once in the chloroplast and in the nucleus, and is involved in stress response in barley. And my PhD student Entila is going to analyze this in more details. But now coming to my talk today. So I want to talk about accelerated domestication. So therefore we first have to say, what is domestication? So here I found one of the notes published descriptions. So domestication, this is sustained multi-generational relationship in which humans assume a significant degree of control over the reproduction and care of another group of organisms to secure a more predictable supply of resources from that group. So this means that we have taken care of other organisms in order to our benefit. And so you might be aware of the start of the agriculture some 10,000 years ago, when the farmers started in order to collect plants or seeds from plants which were favorable for them to grow them in the next season. And here on this slide, you see an overview of traits which the Andean farmers were looking for. So they are almost the same in cereals and in other plants, like there is no fruit abscission, more and bigger fruits, the loss of the day length dependence, the determinate growth, color variation, and of course, much more. And the same more or less happened also in the cereals. So here also the people selected for increased seed number, reduced seed shattering, reduced height, reduced dormancy, and loss of the vernalization requirement. So I think many of you, and usually if this would be in-person lecture, I would ask you if somebody can me who painted these nice sunflowers. So, and most of the people then will reply that it was Vincent van Gogh in 1988. And why I show this here, because this shows nicely and illustrates nicely the mutant variants of sunflower, which were present in the time when Vincent van Gogh was a painter, a famous painter. But recently, there was a paper describing exactly those het phenotypes and identified the molecular bases which are responsible for these phenotypes. And this is at least one of the rare plant genetics papers I'm aware of, where a famous painting is figure number 1. And I want to use this to illustrate that mutations are the driving force of our evolution. And without mutations, we will have no progress. So within this audience, I think I do not need to talk too long about the challenges and the demand for our breeders, as you are aware of the increase of the world population. And as we also, I think India suffers at the moment a lot from heat. So with varying temperature or climate change. So this is the one side, but accompanied with this, so we have also the occurrence and new distribution of, for example, fungal pathogens. So these occur now in places where we have never been detected before. And as usually our breeding material is locally adapted, so this may cause severe losses in the yield. And so the yield stability will be not provided. So what are in the breeder's toolbox? And I used here also this scheme, so which is showing that we started with selection breeding while the world population was still low. And then later on, we did crossbreeding and started then with hybrid breeding because of the beneficial heterosis effect. And then in the 1980s, we came to genetic engineering. So this means that we introduce either endogenous or foreign genes into our plants in order to alter some traits. And last but not least, then also gene editing came into place, where we can now, in a targeted manner, can do mutations in a given genome. And so this is also illustrated here, while the world population is rising. So we have a first, second, and third green revolution, including biotechnology. The problem which occurs during domestication is given here on this left side. So while we started with a quite diverse material, over the cycles of selection, we lost a lot of favorable traits. And so people use different technologies these days in order to increase again the variability in the genomes, in order to increase also to overcome this bottleneck. So as illustrated here, so from landrace to modern elite cultivar, there is a reduction in the also favorable traits which are associated to our landraces. Some more words. What is the challenge for the breeders? And I think the biggest challenge for the breeders is the time. So as illustrated here, so if we take the classical crossbreeding, so you cross your two partners, then you get your progeny and you select those one which has the favorable trait you are looking for. But as the background still contains a lot of maybe an unwanted type, so several cycles of backcrossing has to be undertaken in order to enrich then again the elite material. And this is in usually 2 to 7 years. Then at least in Germany, and I'm not aware how the regulations are in India, but if you want to release a new variety, you have to show on several sites over several years that the new variety outcompetes what exists already. And so this takes, at least in Germany, another 2 to 3 years. So that you end up to develop a new variety is about 8 to 10 years by using classical crossbreeding. Then people started to use mutation breeding. So for example, either we applied chemicals or radiation as illustrated here. In this case, you of course induce a lot of mutations in your background, but as there might be also unfavorable mutations, also here you have to do several rounds of backcrossing in order to come to your favorable genotype. And so here is one example. So we wouldn't have the pink grapefruit if we haven't used mutation breeding. And so as I have a long history in barley, so also many of the today's barley varieties in numbers 307 entries are existing, which are based on this mutation breeding. And you might be aware of this International Atomic Energy Agency in Vienna, based in Vienna, and they have a database where they collect all those data, and you can select for your plants, which we are developing by using, for example, radiation. But if you think that this is now outdated, so also recently, for example, here the banana variety Pirama was released in 2019, and this is also based on such a radiation experiment. So it's still an up-to-date technology, but it takes as long as the classical crossbreeding as we need these many cycles of backcrossing. So then we come to the classical genetically modified organism. So here we use either the agrobacterium strain, a natural mechanism in order to transfer either endogenous or foreign genes into our plant. But this integration occurs random, and we also cannot know the number of integrations we integrate. So that's why then still 2 to 3 years you need in order to make a clean homozygous event, but this is already much shorter than for the before, as this testing still applies. So here we end up maybe with half the time we use for classical crossbreeding or mutation, the classical mutation breeding using the radiation. So then I come to the point of the endonuclease platforms, so which are used for targeted mutagenesis or gene editing. So you might be aware that we have these 3 different platforms. We started with zinc finger nucleases, then we had transcription activator like effector nucleases, and last but not least, we have the CRISPR-Cas technology. So all this has in common that it's a 2-component system. So you have one, which is the navigator in order to find your genomic position, and you have a scissor who induce a double-strand break, because this is what we are interested in. We want to induce a double-strand break at a given position, and using the host endogenous repair systems, we then get modifications. While the 2 upper ones are based on polypeptides which bind to the target DNA, the CRISPR-Cas system relies on an RNA, which is the navigator to find it. And here, the 2 above use a scissor, which is called FOC1. And as FOC1 is only active as a dimer, so that's why you need here 2 units, while for the CRISPR system, we have just 1. This 2-unit system may have a beneficial effect for off-targets, as a stretch of 50 nucleotides might be more unique to a given genome than the just 20 nucleotides used here. But for the easiness of cloning, of course, this system is very favorable, and it has been applied many times. And that's why 2 of the inventors, Emmanuel Charpentier and Jennifer Doudna in 2020, got the Nobel Prize for this technology, highlighting the importance and the revolution, I would say, we have achieved. So, as briefly mentioned, what is the basis of this targeted mutagenesis? So we have our complex, we find the position in the genome, and then we induce a double-strand break. So the cells have evolved several repair mechanisms, and here are mentioned the 2 main repair mechanisms. One is non-homologous end joining, where you just take the 2 loose ends and stick them together. And this, in the majority of cases, is of course error-free, but as again a cutting occur, and the process will just stop when we have a mutation. So that's why at a certain time point we lose 1 or a few nucleotides or insert 1 or a few nucleotides, and then we get altered open reading frame, which usually leads to a knockout of the target gene. The other mechanism is called homology-directed repair. And here, usually a donor DNA is used for repair, so that we compare the donor DNA, which is usually the sister chromatid. So and then the repair happens. But this gives us also the opportunity by providing synthetic repair templates that we can alter a certain gene or introduce a certain sequence in a given position. So as briefly mentioned, so the CRISPR technology is very powerful and can be used in different plant species, and it has been already published a multitude of studies in the different categories of our crop plants. So while we have examples for feed crops and fiber crops, the majority of applications so far is published on food crops, whereas crops for industrial use, oil crops, and also ornamentals. So you can see that this is really a very broad application of this technology. And if you look what the people are still going for, what traits they want to alter. So you remember that this I showed you in the beginning. So if you look, so then it's still more or less the same as our Andean farmers were working on. So the biggest are agronomic value, followed by food and feed quality and biotic stress tolerance. So these are the major things people are working on these days still. But what are the requirements for genome engineering? So on one hand, of course, you need to have a sequence information. And here, of course, we benefit now from this great progress made in sequencing technologies, which allow us now that we even can sequence large genomes like wheat for a reasonable price and in a very short time. Then another critical step is that you need the suitable regeneration systems, which means that you transform a single cell, and out of this, you then have to make a new plant. And this is very challenging still these days. And as I briefly introduced, there are differences in the explants and other things. Then, of course, you need suitable expression systems, and this I highlighted also in the beginning. And there are different methods how you transfer your units into the different cells. And one point is also still of high importance is genotype dependency. So this means if you can transform one barley variety, it does not mean that you can transform all barley varieties. So usually coming to the transfer of the functional gene editing units. So here the majority is based on plasmid DNA or linearized fragments, where you assemble all the necessary elements, like, for example, your guide RNA and your scissor, and maybe a selectable marker gene in order to promote the transformed cells over the non-transformed. Then also you can introduce RNA, or last but not least, also you can introduce ribonucleoprotein complexes. These are pre-assembled RNA and Cas protein complexes, so which then gives you the benefit need to care about the expression system, as this is already a functional unit. So you just need to introduce it into your plant cell and it would be immediately active. So then there are different methods how you can transfer your functional gene editing units. For plasmid DNA, the majority of cases use Rhizobium radiobacter, which you may know as Agrobacterium tumefaciens, but it was recently renamed. Then we have biolistic transfer, which is also becoming very popular again, especially if you think in the terms of homology-directed repair, as you can think of that you need a high number of repair template in order to achieve your goal. But there are also other methods like electroporation or PEG-mediated transformation for protoplasts. Recently, also nanoparticles became very popular, as these also allow to introduce either DNA, RNA, or proteins into cells. So next, I want to give you a few examples for Barley domestication traits where I was involved with. So here you see the 7 Barley chromosomes, and over the couple of years, the people identified certain genes which are responsible for certain traits. And to start with, I showed you that the people selected for increased seed number. And so here, one thing which we analyzed together with Takao Komatsuda from Japan is the so-called VRS1 gene. So you might be aware that in barley we have 2-rod and 6-rod barley. And the cause for this is the VRS1, vulgare rod type 6, gene 1. And the effect of the functional protein is the inhibition or to prevent the fertilization of the lateral spikelets. So in barley, you have always 3 spikelets, and in the 2-rod, just the central is fertile, and you get the grain. So if you have an inactive VRS1, so then also your lateral spikelets become fertile or were fertilized, and then you get free grains. So we started early on by using the RNAi technology, which you might be aware. So this is a gene silencing technology. So this means that you reduce the transcript of your target, but it's not the complete. And so here you see on the left side our wild type. This is our model cultivar Golden Promise. So this is a 2-rod barley. And then we silenced the VRS1 gene, and we got a partial fertilization here in our lateral grains and also some awns. And when the CRISPR-Cas technology became available, so I immediately then performed a CRISPR experiment. And so here you see the result. Again, our wild type Golden Promise, which is 2-rod. And here on the right-hand side, you can then see that we fully converted the 2-rod into a 6-rod barley genotype. I removed the awns that you can better see that we have fully developed grains here in this case. And so in theory, you can triple the yield per square meter, but this of course is not true. So as all these grains needs to be feeded, so this means you have also maybe to alter the allocation of amino acids and other stuff. And as the head is more heavy, so you may also have to interfere with your stem, as these might be more prone for lodging. But for the first time, we can really analyze the agronomic value of this gene, as all natural VRS1 or all natural 6-rod barleys may have other mutations in their background which are undiscovered. So here, the difference between this and this plant is just a single nucleotide. The second one, which is of course also one of the domestication traits for barley, is the reduced seed shattering. So you might be aware that the wild relatives, of course, were brittle. So this means that they lost easily their grains in order to become distributed. But this, of course, was not favorable for our farmers. And again, with Takao Komatsuda, we showed that, or we identified and characterized the brittle genes in barley. So naturally, the domestication of this brittleness phenotype occurred twice. So we have 2 genes or proteins which are responsible for this effect. And if one of the 2 is mutated, so then you keep your grains in the spike. And so on one hand, we have one base pair deletion in the BTR1 gene, which makes the phenotype. And then we have another deletion of 11 base pairs, which is in the BTR2. And so both brings or let the grains stick to the spike. And so we confirmed that we identified the correct genes, and this has been published, as you can see. So Golden Promise, the model cultivar in barley, is BTR1, so small BTR1. So this means has a mutation in the brittle 1 gene. And so therefore we complemented barley Golden Promise with the functional brittle 1 gene. And then here you see the outcome. We converted a lead variety into wild barley. So because the spike becomes brittle. And we confirmed this also for BTR2. So here we used the recombinant inbred line, which we were luckily could transform. And so here, by introducing the functional BTR2 gene, then we got the same effect that also this spike become brittle. So we could confirm that those genes are the responsible genes which keep the grains in the spike. Another one, which is also related and mainly maybe in China and in the Nepalese area, very used. So you might be aware that we have also naked barley. So here, usually the lemma and palea stick to the barley grain in the non-naked. And here, together with Sofia Gerasimova, we have shown that the nut gene is responsible for making this phenotype. So Golden Promise usually is not a naked barley, but by making a mutation in the nut gene, then you get also the naked barley, and this might be or is used in parts of the world, so as kind of snack. So in Europe, we usually use sunflower seeds, but I have been to Nepal, this part of China, so we are the Tibetan highland, to be correct, and where they sell it on the street, so you can eat this naked barley. I don't know how it's in India. So then a problem which is not relevant for Europe, but highly relevant for Japan is reduce in dormancy, as where, because of high humidity, the grains already start germinating while they are still in the spike. And so here also the group in Japan used first RNAi to show that the QSD1 locus is responsible for this effect, so which is the gene for dormancy. And so then my colleague Robert Hoffi made also then mutations in the wild type, so which is Golden Promise. And then here again, you see that those also have a reduced sprouting or germination. So in theory, you can, by using this technology and by knowing the genes which are responsible for these traits, you can convert wild species or a landrace into an elite variety by using this CRISPR-Cas technology. So this I find very attractive, and this would speed up the breeding process, as we can make multiple edits at once. And so this would be, within a single generation, we can convert such a landrace into a modern elite cultivar. And this is very attractive and amazing. And this, of course, does not attract just me. So there are already published studies. So here, for example, one for the novo domestication of rice. So here, these scientists, they altered wild allotetraploid wheat rice into domesticated diploid rice. And another example is given here. So this is for tomato. Here, the people, by altering 6 genes, which are involved in the fruit size, the fruit color, and the composition, and in the side branching, and in the height. So we also could convert in a single step this plant into this very compact and very agronomic relevant plant. And the same genes has been also used in order to alter ground cherry. Of course, the orthologous genes has been targeted. And also here, we could see the same effect, that we could alter this in one single transformation. And all those things are published. So, and with this, I would like to end. I hope I could show you that using the CRISPR technology, so we can really convert species into a lead material, and we can introduce so also new traits very quickly, as this is highly demanded. And so here you see my collaboration partners at HHU, at the University of Cologne, and of course many of the studies I showed you today has been performed in my former group at IPK. I thank my funding bodies, and I thank you for your attention, and I would be open for questions.
Thank you very much, Hensel, for a very elaborative talk, and the way you spoke. I think even the persons who are not involved in this CRISPR, they can understand your presentation, your idea, and your research, whatever you have related. Sunil, can you unmute yourself? Sunil Archak, can you take up the questions which have been asked?
Now I am able to unmute myself. Thank you very much.
Just read out the questions.
Yeah, I'll do that. Thank you, Dr. Hensel, for an excellent and captivating, full of nice graphics, very easy to understand for everybody. Thank you very much. There are a couple of questions. As you perhaps are aware, Dr. Hensel, recently India has brought out guidelines for, let's say, CRISPR modified plants for public use. So there are various questions. Half of the questions will be about the technology per se, and half of the questions will be about the challenges that one is going to face when somebody is using these technologies. So a couple of questions are already there in the chat box. I'll read out for everybody's benefit, and then I request you to answer one by one. The first question is that when you started working with, perhaps this was related to your barley phenotypes, Dr. Dinesh Kumar from Indian Institute of Oilseed Research is asking, did you find differences in the phenotypes when you edited different parts of the same gene?
This is, of course, a very relevant question. And so the answer is yes. So because this might be also one thing which I always would recommend if you design your CRISPR experiment, that you look carefully where you have maybe alternative transcriptional start sites. So this means usually people think if you have 5 exons, you make a cut in the exon 1, then you abolish your protein function. But there are examples. For example, we have a lipoxygenase gene, for example, which is responsible also for the foam quality in barley beer production. So here you have an alternative transcriptional start site in exon 5, which gives you an attenuated protein function so that you still have some LOX activity. Also, you may have a mutation in exon 1. So you get a reduction, but still not a complete knockout. And so that's why you should always carefully check if you know where you have your protein domain of interest, that you make the knockout there in order to really be sure that you get a full knockout. And also maybe highlighted by this, and so we have also examples in Barley that by using this technology, you can also get such an attenuated protein function by using two guides and cutting out some nucleotides in between. Yeah, so that you have a -3, -6, -9, that you delete certain amino acids. And so thereby, of course, you may change the structure and the function of a protein. And this would somehow mimic the RNAi effect, where you get the silencing. But for the RNAi effect, you always have to overcome a threshold. And here it's genetically fixed. So this means it will be always applied. And so with this, so if you have maybe an essential gene where a complete knockout would be lethal, so then by just deleting certain amino acids may still give you a relevant information, but a different phenotype. Yeah, so that's why full knockout would be lethal. If you may omit 4 or 5 amino acids, the plant still survive and show an altered phenotype compared to the wild type. So yes, clearly yes. If you have mutations in different positions of the same gene, you can expect also certain or different phenotypes. Yes.
Yeah, that's excellently. Thank you very much. Dr. Hensel, now people are also working on CRISPR-Cas based experiments here in India, many labs. Everybody has one common question, that is, how do you, what is the frequency with which you observe, let's say, off types? And then that's first part. Second part, do you document the observation of off types, and then how do you manage that? Off types.
So you mean off targets?
Off target.
Yes. So of course, so in any place we are talking about CRISPR, these people are asking the questions about the off targets. And I think in the human and medical fields, this is of course much more relevant, as in the breeding process. If a plant looks somehow weird, you just throw it away. But this is of course not a serious answer. So of course, we want to have as less as possible off targets. And so therefore there are certain online tools which you can use in order to select your guide RNA, which then also double check that the 20 nucleotides which you use are unique for your genome. This might be very challenging for larger genomes. And so therefore then you can also look for the number of single nucleotide polymorphisms, so like mismatches. So it has been shown that the 12 nucleotides in front of this palm, so which is necessary for the binding of the protein and which coming downstream of your target sequence, that if there is a mismatch in these 12 nucleotides, that you do not expect any off target. So that's why there are online tools which also give you those predictions, how many mismatches you have in your 20 nucleotides in another place in the genome. And then I usually look into the sequence if there is a mismatch. Of course, if there is no mismatch, then this would be a serious target site you have to consider and to check. And we have examples in plants where we have on target. So this means in the position we want the mutation. We have also mutations in an off target. But we have also in the same experiment plants which do have just on target mutations. So, and then of course you have to carefully screen your plants for this effect. Yeah. So if you have mismatches, my advice would be to check at what positions those mismatches are. If they are towards the end, so the 5' part of your guide, so then they can also be tolerated and you can still expect off target. So which is also indicated by the fact that also 18 nucleotide long guides are effective. Yeah, so this already indicates that if you have a mismatch in position 19 or 20, will be tolerated without a big decline in your activity. So that's why, yes, I use usually computer programs in order to predict my off-target positions. Then I check if I have a mismatch, and if this mismatch is in a relevant position, as I explained, towards the 5' end of the guide, then of course I design primers and check these positions as well.
Yeah, thank you. So I may just like to ask a related question. Yes, we understood the technical part of that one. Now about the standard operating procedures, let's say in the lab. Is it means how hard and fast or how specific it is to record these off-target ones? Because as you just said in the beginning, all you have to do is to just remove, because that's not required. But how do you, how much essential it is as part of the standard operating procedure to document the occurrence or the numbers of these off-targets, and then what is the part of that? How do you go further with that?
So initially, we of course just screen our plants for the on-target and look for the respective phenotype. And then if we have a number of plants which have a on-target mutation and show the phenotype, or not saying just expected, yeah, of course, if you find in a certain number of mutants a certain phenotype, then of course you have to make sure that this is just based on the mutation you introduced. And in those plants, so in order to reduce the workload, of course, we don't screen routinely all our events for off-target mutations. We just do it for a certain number of plants where we have confirmed on-target activities.
Okay, okay. Thank you. Now that one question I should ask about the which pertains to the title of this particular talk, and that is my colleague from Indian National Gene Bank is asking Dr. Dharma Prakash, do you see any epigenetic changes after gene editing in different plant systems? If they are not reported, what is the possibility? How much is the expectations?
So we do, of course, not routinely check for epigenetic changes. And so that's why I cannot really give a serious answer to this. So I can just mention that we haven't observed in the plants we have generated an epigenetic effect, which does not mean it does not exist. But we have not checked for it. So I'm sorry, I can't answer this from my own experience. Yeah? In our experiments, we did not observe any epigenetical changes, no obvious. I have to say no obvious. Yeah? It does not mean that there is not an epigenetic effect, but it's not obvious.
Yeah, thank you. There are a few more questions, but your email ID has been shared with the participants, and okay. I hope you would be happy to respond to them directly.
Sure. Sunil, let us take one last question with Dinesh Kumar.
Yeah, please.
Okay. So you can read the Dinesh Kumar's.
Yeah, that Dr. Dinesh Kumar has clarified on his first question, sir.
Okay.
But, yeah. So I would rather have him directly contact Dr. Hensel, because that's a little detailed question on silencing part.
Oh, yes, the second question is, what are you suggested strategy to adopt genome editing approaches in crops that are recalcitrant to in vitro manipulations?
Yeah.
Sorry, can you repeat one more time, please?
I'll read that one.
Yes,
what are your suggested strategies to adopt genome editing approaches in crops that are recalcitrant to in vitro manipulations?
Of course, yeah, a good question, and of course very challenging. So we have also shown that maybe you can also use white hybridization. So this means For example, you can pollinate a wheat plant with a maize pollen. You get a fertile fertilization, and then during the course of the further development, you omit the genome of the one elder. So in this case, the maize or the corn. And so as even wheat transformation is very challenging, and I also briefly mentioned the genotype dependency. So then you can use these wide hybridizations for genome editing as well. So we have shown, as we were easily able to transform corn, so that you need just one transgenic corn plant, which has a CRISPR construct, which targets wheat genes.
Yeah.
So, and then, of course, it should not make any off-target in the corn. So this you have to make sure by the selection of your guides, so that they are wheat-specific. And then you pollinate with those pollen different wheat varieties. And so we could show that we could achieve targeted mutagenesis in bread wheat and also in pasta wheat, so in tetraploid wheat, by using this technology. Yeah, so at no stage the wheat plant is transgenic, so you also overcome this problem of transgenesis, and also to get rid of the tDNA, which we usually have. Yeah, so of course I did not had time to talk about the application of these RNPs, which we also use in order to overcome that you have to segregate out your CRISPR transgene in order to avoid also in further generations some off-target effects. Yeah, so that's why usually after we identified in our primary mutants mutation, then of course we select in the progeny those plants which are non-transgenic but still carry the mutation. And so here, by using these wide hybridizations, so you then do not have just in the initial state transgenic cell, but later during the development of the wheat plant, there is no transgene at all, as the transgenic corn chromosomes get eliminated. And so in this case, you have just a mutated plant, and this works for any genotype which you can pollinate with corn. And so there are other similar systems like Barley with Fordium spontaneum, bulbosum. So there are other those systems where you can use it. Then, of course, people are also working on other methods to overcome this in vitro culture system. And so there are, of course, also the use. So the main problem is not the transfer of the unit, so usually the regeneration. And here are also methods available that you co-transform, for example, WUSCHEL or GRF-GIF in order to make new meristems and to get new plants out of your tissue.
Okay. Yeah, I think that helps the person who has asked the question understand a little more than what has been already mentioned. Thank you very much. I just hope, Dr. Hensel, that this is only the beginning of the interaction between Indian researchers and yourself and your lab. And we expect that the interaction will grow much more into
many thanks.
Collaborative projects and people visiting your lab and working with you and then understanding and then working together on this very important topic. Thank you very much. Dr. Agrawal, please.
Thank you very much, Hensel, and thanks to Dr. Sunil. Thanks to our all colleagues who have joined for this important lecture on a very important topic. As we could know from Mr. Hensel, that what's the latest going on, and he very nicely elaborated the various concepts of the breeding, historical, taking the historical aspects, and then to the modern time. That's really very educative, and some of the scientists would like to maybe collaborate with you also. I have shared your email ID with them.
Many thanks.
Yeah, so thank you, Hensel, for this important talk and for responding to the queries of our audience. who are working in the field of biotech, this CRISPR and genome editing and domestication. Many organizations which are dealing with this, they have attended this lecture today. So I think that will really greatly benefit this. And your lecture has been recorded and it will be made available on our Indian Council of Agriculture Research website. So people and the students can take advantage even later on. So that's another advantage. So I think it has been a very great talk. And thank you once again. And thanks to all the audience for attending this lecture and putting your questions to Mr. Hensel, to Dr. Hensel. Thank you. Thank you all. Thank you. Namaskar.
Many thanks. It was a great pleasure.
Thank you
for being here. So have a nice day.